Welcome everyone once again. Welcome everyone. So today the first section will be taken by Mr. Sanjay. He's the Associate Vice President of Agappe Diagnostics International. He has over 20 years working experience in the IVD sector. And so he'll be taking us through the importance of the Agappe MISPA I3. So Mr. Sanjay, over to you.
Yeah. Thank you, Kimi, for the smart introduction. I believe that my screen is visible to all of you.
No, okay, yes, we can see your screen.
Yeah, just a minute. I think. Yeah. So, first of all, thank you very much for joining this webinar. And I'm Sanjay, the Associate VP of Agappe Diagnostics. And I know that, there it is good evening, and here it is, there may be good afternoon, and here it is, good night time. It is 9 p.m. In Japan. So first of all, thank you very much for giving us this opportunity to interact with the senior teams, senior technical teams in there. So we all aware that the diagnostics or diagnostics are actually increasing like anything, right? So if you look at the evolution of biochemistry systems in the 1980s, it was a manual era where all the tests are performed manually, means whether it is semi-automated system, whether it is photometry system, all those things are manually performed. So the reason is that there is not big systems are available for the common population. When it comes to the 1990s, the semi-automated systems actually taken place of the the manual methods, like I would like to say the photometer to semi-automated systems, and in 2000s it all give way to fully automated systems, and in 2010, 2 decades ago, it becomes systems with connectivity, and in 2020 onwards, actually today the system is intelligent systems or AI based systems. So there is a huge jump, or I would like to say in a huge jump in technical advancements when diagnosis and diagnostics are concerned. So over 3 decades, say when I started my career in the year 1999, so when we performed doing some tests, some of the tests we still used at that time, 98, 99, that time, we still used something called as, photometer, something called as, spectrophotometer, and some system which actually uses glass, glass slides or glass filters. So that era is already gone. Today, everything is in fingertip. Everybody is using automated systems, from keyboard system to touch screen system. Now there are systems which can give you a voice prompt. Now, if you look at the global case studies of today, that is the automation driving clinical excellence, 52% of turnaround time can be reduced when we use the automated systems of the newer segments. There is 89% decrease in the errors, what is happening. And in the throughput, if you look at, it is increased by 3.2 times, and annual cost savings is considered to be around, average is around 2.4 million US dollars. Use automated system. These are some of the examples taken from Singapore, in Berlin, and Israel, etc. So where it gives much importance to the automated system. You are aware that there is something called as control materials, right? In all processes, there are three phases in the testing. One is pre-analytical phase, post-analytical phase, and post-analytical phase. The two phases, we have control. We have control mechanism. Example, analytical phase. In analytical phase, we have control mechanism. The control mechanism is running controls, QCs, participation in proficiency testing, IQCs, EQA, interlab comparison, all those things are there. So we can control that process. That is why if you look at the analysis, the errors that is happening in analysis is only 8% to 9%. When you look at the pre-analytical phase, there are now automated systems are there. Then maker checker processes are there. So there also the error will be around very less, maybe 18%. Whereas if you look at the pre-analytical error, still around the world, the pre-analytical error seems to be the highest, which is more than 60%. So out of 100 errors which is happening in a laboratory, more than 60% of the error happens due to pre-analytical variation. But okay, that is apart. Why we cannot control that? Because it is performed by human. So in human, we need training, we need CMEs, all those things. So these are the three phases and three types of errors that is happening. Now, when we look at the principles, there are different types of principles today available in the IVD segment. The first one is photometry. We all know that what is photometry, the basic principles of photometry. The second one is something called as a nephelometry or turbidometry. Turbidometry is also there and nephelometry is there. Nephelometry is nothing but scattered light. Yet photometry is, satisfy only the transmitted light, whereas the nephelometry, it satisfies the scattered light. Another is something called as, counting technology, that is cell counting technology, counting. It is only for counting. We call it as a Coulter principle. And other one, we can say it is something called as, flow cytometry, which is used to identify different cells. That is called a flow cytometry. And the third one is a common one called as a CLIA or chemiluminescence, or I would like to say ELISA or immunoassays, like, ELISAs, radioimmunoassays, chemiluminescence immunoassays, all those kind of things. So these are the different types of principle or detection principle which is used in the diagnostic field. Now, when we look at the three pillars, the three pillars challenge is nothing but accuracy, affordability, and accessibility, 3A. So which means whatever instrument we are going to have today in the diagnostic laboratory, number one thing is that it should be accurate. I believe that everybody sitting in this webinar will accept this point. The second is affordability. Yes, you have a machine which is very good or it is accurate, but you cannot afford it, which means you are eluding the common population from good, I would like to say good, diagnosis. The third one is accessibility. What is accessibility means? Accessibility means it should be a compact system. It should be a robust system. It should be accessible or it should be addressable. I can even say addressable to the general population, the medium population, and the higher population, which means it should be a cosmopolitan system. So these are the three things we should understand as a requirement when you consider the diagnosis world of today's market. Now, I said that accessible, accuracy, affordability, and accessibility, all those things. Why? Why? The reason is that today, if you look at specific protein testing is actually, increasing in the world. When I was performing test in 1998, that time, the most common test was CRP and RF and ASO. These are the three parameters: CRP, RF, and ASO. Prealbumin, transferrin, captoglobulin, ferritin, C3, C4, immunoglobulins, all those things are considered to be specialty tests. So which means we collect the samples, then the samples are kept and the samples are sent to a reference laboratory to do the testing. Sometimes the reference laboratory also, they will collect all the samples, they will pool the samples, maybe alternate days, sometimes C3 and C4 they will do twice in a week, or haptoglobulin once in a week, or prealbumin once in a week, like that, which means that we need to wait for a week's time to get the results. So lot of uncertainty for a period was there. So that is 2 decades ago. But you know that now today, if you look at all over the world, the specific protein testing is going like anything. The reason is that it will give It will give you very specific answer or very specific diagnosis. Everybody know that, say for example, CRP, if an inflammation is there, CRP, SIA and ferritin will give you an idea whether what kind of inflammation is there. Say for example, ferritin. Ferritin is one of the sought test in in during the COVID time, where the ferritin is used as a measure, as a diagnostic tool to understand whether the patient is having septic shock or undergoing septic shock. So every specific protein is having its own importance. So, but the question is, oh yes, the specific proteins are having its own importance. Are we capable of doing this? Is it possible to do the specific protein in a medium setup or in a smaller setup? The reason, maybe in 2 decades ago, it was actually a no, because the hardware which is used that time, it is of costly affair. And second thing is that, the consumable cost or the recurring cost is very, very, very high. So these are the reasons, for not performing these tests, in the earlier time. But of course, there are machines. The machines are very good. Only thing is, it is accurate, or it doesn't have an affordability, or it doesn't have an accessibility to the common population. That was the issue. Now, today, the protein estimation is gaining popularity. The reason, from 2010 onwards, there are different companies or different products came into the semi-automated to medium automated segment. There are immunofiltration techniques. There are lateral flow techniques. There are nephrometry techniques. There are turbidometric techniques. There are different systems actually evolved. I would like to say from 2010 onwards, around 15 years ago was the evolution. I would like to say, even in that time, there was only smaller machines available, very smaller machines, very smaller one or two companies are hello.
Hi, we can hear you.
Can you hear
Can
me?
hear you now. Yes, we can hear you
Yeah,
now.
but the slide is gone.
So we can still
Can you hear
see
the
your
slides?
slide. Yes, we can still see your slide.
Okay. Okay. Now, today, if you look at, there is a shift from curative to preventive healthcare. If you look at today globally, now today I'm in Japan. So in Japan, we are having some projects. So here also, everybody is thinking about curative, instead of curative to preventive healthcare. So there also, they are talking about how we can prevent the disease, or how you can early diagnose the disease. So from reactive mode to proactive mode, I would like to say. So where they are here also, they are, instead of making bigger system, they are thinking of smaller or compact system to address the market demand today. Now I'll give you a typical example. Earlier days, the manual techniques, you can see that it is a CRP testing. Okay, the CRP testing, when you look at manually, if it is positive for multiple titration, say for example, if it is positive for 1:32 titration, the value comes to 192. When the same thing is positive for 1:16 titration, the value comes to 96. But so, which means 96 to 192, there is a huge jump or a huge gap. This was unable to identify whether the patient condition is coming good or the patient condition is going bad. This is the reason people started from manual to turbidometric or to nephrometric methods. Now, look at here. Earlier, this is for ASO. Earlier, it is plus, minus, plus, sorry, not ASO, it is for RF, minus, plus, plus, triple plus, 4 plus plus. These kind of gradients was also there. Here also, the problem lies with the person who is actually reading. So these are the limitations actually which happened in the earlier days. So when it comes to preventive medicine, what we require something which is more accurate, which is more precise, which is more affordable, and which is actually giving some value addition to the person who is doing things. Now comes, see, the one of the reason in the earlier days is that, please understand, latex-based assays. This latex is nothing but a sphere. It is a sphere, spherical shape. It's something like a football, I would like to say. In the football, there are many antigens or antibodies coated. I'm just giving an example. So what happened when two antigens or two antibodies come together with when it collide, it will form a cluster. I'm just giving a simple example. So earlier days, the latex particle size was very little bit higher so that we can actually look into the slide or we can see through the slide. So it was having a problem with accuracy. But in the later 2010, the latex particle was very small. Then came something called as latex enhancement. That process is called as latex enhancements. That is why those assays, in that time it is called as LEIT assays or latex enhanced immunoturbidometry assays. Why immunoturbidometry? Because an antigen combines with antibody to form an antigen-antibody complex or a turbid complex, and that turbidity is measured with the help of latex particles. That is why it is called as a latex-enhanced immunoturbidometry assays. Now, these immunoturbidometry assays using the latex particles or LEITs actually required not a turbidometer. It required a nephrometric principle, because here it is not the transmittance what we measure. It is actually we are measuring the the scattering. So since the scattering is measured, so this era, that is 2010 onwards, the rise of protein systems become evident in the market. I would like to say 2010 to 2020 is the rise of protein systems. Again, we can say like that, rise of protein system. Turbidometry, the issue is that, it is having its own merits and demerits, but still there are systems which can actually have both both turbidometry as well as nephrometry. Now, what is the limitations of any immunoturbidometric reaction? It is nothing but Poisson effect or Hooke's effect, which means that if antigen is excess or antibody is excess, what happens? The reaction won't be proper. Where there is something called as an equivalence zone, where the reaction will be very maximum. So if antigen is excess, what will happen? The reaction will come down, which means it will give a false negative reaction. So this reaction, that is the antigen antibody reaction, is not a linear reaction. After some time, it will actually come down, even though the antibody is there, but because of the excess antigens, or even though the antigen is there, because of the excess antibody, there will be some non-linear reaction which is happening, which is called as the Hooke's effect or non-linear reaction. Because of that, what will happen? See, like say for example, glucose. If you add more glucose, what will happen? The reaction will go like the straight curve. So you will get a linear graph. But in this case, if the antigen is excess, instead of getting a high precipitation, the precipitation will come down. So here comes something called as a combination or dual channel technology. Again, from 2010 to 2015, I would like to say it is a rise of protein systems. From 2015 onwards, I would like to say instrument with the dual channel technologies and with the dual measuring principles came into picture. Now, when we look at that, please understand there are some factors which actually creates good accuracy in nephrometry. Number one, the particle size and shape. How good is the particle, or how small is the particle? Then the wavelength which is used, usually for scattering, the wavelength used is 630 onwards, because it is in the infrared region. Then distance of observation, which means where the cuvette is kept and where the light is there, because the distance from the setup actually it gives something called as the reactivity, then the polarization, then the concentration of the particle, and also molecular weight of particles. So these are the factors that affect the light scattering. So based on this, in the 2020 onwards, no, not 2020 onwards, 2015 onwards, the next generation of analyzers came into the diagnostic field. You can see the symbol scattering pattern. When the particle size is small, when you just pass a light into the particle, the scattering will be uniform. When it is a large particle, the scattering won't be uniform. When the particle size is very high, the scattering will be highly irregular. So based on this and based on this, the scattering pattern, actually most of the current specific protein systems are working. So this is the new generation specific protein systems. Now look at this. In common, automated systems, to do a calibration, as I told you, all those specific protein assays are non-linear assays. You require something called as a double fold dilution, which means you need to dilute the calibrator one after one after one after one after one after others, and you need to plot a graph from the calibrated or from the diluted calibrator. This was one of the constraints for many customers or many technical staff to adopt certain specific proteins, because even if any of the point there is a failure, they need to repeat the test again. So it is a problem to them. So what happens is that, no, linear to non-linear curve, there is a point I would like to say, it is a decision point. We would like to say the decision points. So many times this linear to non-linear decision point making is a constraint to most of the technical stuff. So because of that, there comes the technology that is UCS technology in the protein systems. The difference is the new generation specific protein systems use the rate nephrometry, where the rate of reaction, that is, one is taken in the photometric region, another is taken in the nephrometric region, and based on the rate of reaction at a particular point of intersection point, it will decide whether it is a good reaction or a bad reaction, and based on that, an algorithm is paid, and based on the algorithm, the results are made, and the results are more accurate than the earlier systems. Now, if you look at this, Agape in this context, we have two systems right now. One is the I2 and another is an I3. I2 is semi-automated system and I3 is a cartridge-based system. Both the system uses the latest, the patent, I would like to say patented. I'm happy to say that, it's a patented technology. We call it as a UCS patent. Then more than 25 parameters are there. It is a compact system and cost-effective testing. Now, if you look at this, the importance of this thing, I'll just tell you. See, it's a case study. It's an old case study because of which actually we thought that we will come up with something without calibration. Now in our system there is no calibration is required. The reason is that, see, look at this. In these two curve, in the first case a patient got 5.74, which is not what do you call it, positive. On the other case it is negative, on the other case it is positive. The reason is very simple. In the second time, one of the point, the calibration curve is not good. See, point number 5 and point number 10, calibration point number 10, the OD was mismatched, because here the OD is 0.033, here the OD came 0.020. Because of this calibration was a problem. Look at this. We cannot simply identify whether the calibration failure is there or not. So this is the reason we came up with an idea. Why can't we provide calibration also in a smart card? So that is the birth of system with a smart card calibration. Now, I believe that most of you know this product. It is available in 60 plus countries with the 7,000 plus satisfied customer as of now. Now, yeah, this is the portfolio, parameter portfolio. See, one thing is that the system, whatever we are using, it is having, single cartridge, one cartridge or one test. Okay, no need for taking the reagent, no reagent deterioration, no reagent stability issues, nothing is there, only single cartridge. And you can say that here around 25 different parameters, including ApoA, ApoB, free kappa and free lambda. This is, I would like to emphasize on free kappa and free lambda. You can see that immunoglobulins is there, cardiac marker is there, kidney marker is there, then what you call the other lipoproteins are there, complement assays are there, diabetic this thing is there, inflammatory markers are there. Of course, another one, then vitamin D is there, then deep vein thrombosis, that is, D dimer is there, and the more important is that FLC, that is free kappa and free lambda is there. And I believe that, there is no system right now available in the world with this kind of a extensive menu, including free kappa and free lambda. See, it is, as I told you earlier, today what we require is a preventive medicine. So in preventive medicine, lot of preventive checkups are there. And in preventive checkups, people want to understand whether I am prone to any particular disease. So how we can find out whether I am prone to a particular disease? Today, there plays an important role by specific protein. Now, look at this. Look at this particular, this is taken from one of the clinical studies. Okay. So here, multiple myeloma. I think everybody knows about multiple myeloma. Multiple myeloma is one of the fastest spreading plasma cell disorder in the world. And you can see that even in African continent, even in Indian continent, across the globe. And it says that more than 180,000 multiple myeloma cases are diagnosed each year globally. And out of that, around, the highest is in, US with 35,780 multiple myeloma cases, followed by, some other countries. And this particular disease is prevalent in the African continent also, where, 3 to 4 out of 100,000 people are prone to this disease. And another thing is that, age is not a barrier, and, the sex is also not a barrier. Now, there are undiagnosed cases available in many of the countries. The reason is that this. If you want to know if a patient is suspected of multiple myeloma, then the usual test today, what we do is an electrophoresis or a serum electrophoresis to understand the M-band. You can see that on the left-hand side, there is, a protein electrophoresis, electrogram, where you can see that, the normal electrophoresis pattern, where, it is given in blue, whereas in multiple myeloma cases, you can see an M-band, or on this side, there will be an M-band. So this, along with the, PET-CT or positive emission tomography, gives an idea that yes, the person is suffering from multiple myeloma. But dear friends, it is understood that this protein electrophoresis or PET-CT scan is not available everywhere. So for example, in my city, where we are having very big hospitals, but PET-CT is not available in all of those big hospitals. Only hospitals which is there, which is having a very good oncology department, is having the PET-CT. And even today, for the electrophoresis, many of the laboratories actually depends on the reference centers. So this is one of the constraints. So here comes, it is about free light chains. So it is nothing but in plasma cell is a type of cell where, normal light chain will be like this, whereas, in case of multiple myeloma patients, the light chains will be free. Some light chain will be free, and this light chain will give rise to something called as a plasma cell disorder. So, and it circulates in the blood, and in most of the multiple myeloma cases, we can see abnormal cells or abnormal plasma cells in the bloodstream. So these are simple, notes about, what is a free light chain and what is a bound light chain and all. So the importance is that if the FLC is there, or FLC, it is not like that, free light chain and free kappa light chain and lambda light chain you are detecting and you are throwing. No, it is not like that. It is the ratio between kappa and lambda. There is some guidelines. The physicians or the referring doctors knows that. So if there is a positive thing, it may be due to multiple conditions like amyloidosis or lymphoproliferative disorder or MGUS or even multiple myeloma or solitary plasmacytoma, or else it is something called as Vanderstrom microglobulinemia, a rare type of blood cancer. So actually we are detecting the carcinogenic cells or the carcinogenic cells well in advance. This is something we should understand. See, it says that a patient is detected false positive is okay, because the patient will be tested again. But a patient with false negative is always a problem, because he will go undetected. So with um, with the help of systems like I3 systems, I'm not telling only I3 or similar systems, we will be able to do the test like free light chain or FLC or kappa lambda free light chain, which can help to identify the multiple myeloma on an earlier stage. Now, another is atherosclerosis. If you look at around the globe, there are cases where the person may be healthy individual, no issues, no previous histories, actually prone to attack, cardiac arrest, or similar incidences. The reason is that the bad cholesterols like ApoAs, ApoBs, and LPAs or lipoproteins. If you look at this slide, you can see that even though his HDL and LDL values are within the acceptable limit, because of the ApoA, ApoB, and LPA or lipoprotein A, there is always a chance of getting this kind of issues. And LPA in the medical fraternity, it is called as a silent killer. Actually, it will not show any symptoms like that. It is actually responsible for the myocardial infarction. It is responsible for the ischemic stroke. It is responsible for the atherosclerotic stenosis, and it is also responsible for aortic valve stenosis, which are all fatal cases which will give rise to issues for the patients. And LPA is considered to be a silent killer. Now today, if you look at there are hospitals, or there are laboratories, or there are standalone and chain laboratories who do not the lipid profile as a whole. They do lipid profile including ApoA and ApoB, so that any such cases can be detected well in advance. Now, that is about all those things. Now, when we look at Agappe and Uni-medical, we are a partner for so many years. Now, they are very good. They are having some, it is an ISO 9001 2015 certified company. They are having one of the biggest, I would like to say consumable sales globally. And Agape, we are having 30 plus years of healthcare innovation, and we are having presence in 90 plus countries worldwide with deep understanding of clinical workflows and robust service and support infrastructure. And with the combination is these two, we are actually going to provide, or we are providing one of the best services available for the country. Now, when we look at the future, beyond 2026, now today you can see that I'm wearing something called as an a ring. You can see the ring. This ring is nothing but a wearable device. This will actually give me what was my heart rate, what was my SpO2 rate, what was my temperature, and also it check whether my calorie burn is okay, my steps are okay, all those things are okay. What I'm trying to tell you is that now non-invasive diagnosis is actually evolving like anything. The best example for non-invasive diagnosis is nothing but the pulse oximeter. During the COVID, I believe that every family in the world might have purchased one pulse oximeter to see whether the SpO2 is okay or not. Even I purchased four units for my family, something like that. So the future of diagnosis is very bright. Now, there was a paper, last month, there was a paper where, people were talking about, non-invasive bilirubin for the kids. Now, another thing, I believe that, most of you might have heard about something called as an LOC or lab on chip, which means for one drop of blood, you can do many parameters. Earlier, we thought it is something, in the science fiction movies, but today it is possible. Even we are having a system where from one drop of blood, you can do around 19 to 23 parameters. That is from 100 microliter, we can do 19 to 23 parameters. So things are evolving like anything. Then another one, continuous monitoring. See, earlier, how we monitor glucose, the glucose monitoring is nothing but maybe a glucometer. Once in a while you do, or else you do HbA1c, that is for 90 days average, or else you will do something called as, fasting or PP sugar, like that, right? Now, today, there is continuous glucose monitoring system or something called a CGMS, which will always give you an alarm. Okay, your blood glucose is going high, going low. That kind of system is already there in place. Another one is that, point of care revolution. Now everything is going point of care. Everything is going on like a small system. You yourself can understand that earlier when we are having when we are started our career, the systems are bulky system. One fully automated system means half a room. Now today, automated system from half a room with the 120 to 180 tests per hour. Today, it came to the size of 2 or 3 laptops, something like that in size, I'm telling. And it becomes a benchtop system, automated benchtop system. So evolution is actually happening in here. Another thing is a molecular integration. Molecular integration means, now earlier we were thinking about, whether it is good, bad, but today there are definitive diagnosis is possible, like with the molecular testing like NGS or next generation sequencing. We are able to identify the real factor, or we are able to confirm, yes, the person is having some diseases or not, or confirmatory tests are now becoming popular. And another one is predictive AI. Now everything is predictive AI. So predictive AI is actually coming into picture. So with that, if you look at tomorrow, the next decade will witness diagnostics evolve from a reactive service to a predictive service. We are not just detecting disease, we are preventing it, personalizing treatment, and fundamentally extending human health span. So this is going to be the future diagnostic beyond 2026. And I am in here in Japan for that kind of a project. So that is why I'm very proud to say that this future of diagnosis is very bright for all of us. So this is my last slide, and thank you very much.
Thank you very much, Mr. Sanjay. That was really very informative. And one of the key things that we're taking away from there that really concerns or speaks to Nigerian healthcare is the 3 A's. We want healthcare, or want medical equipment that are affordable, accessible, and are accurate. And it's a good thing to note that our MISPA I3 fits into all 3 categories. So quickly, we would be introducing our second speaker in person of Professor Adedeji. He's a chemical pathologist and consultant at Obafemi Awolowo University. So he'll be taking us through from delay to diagnostics and explaining how smarter diagnostics can transform patient healthcare in Nigeria. So he'll be talking about the importance and the impact of diagnostics delay in patient outcomes, and also stressing on how we can improve or mitigate these problems, especially in the Nigerian healthcare system. So, Professor Adedeji, the floor is yours.
I'll be saying that it informs medical practice. Like more than 70% of medical decisions depend on laboratory data. And recent advances in technology and economic consideration are driving a very high increase in laboratory testing productivity and efficiency. And in recent years, the former concept of already being a specimen center provider of results, just provide results, just produce results, is being transformed into a patient-centered care that determines the final outcome in management. This interaction between the patients as first elements, the physicians, the clinicians are the second element, and the clinical laboratory have been conceptualized to be called brain-to-brain loop. And we're now looking at how smarter diagnosis can transform this patient care in Nigeria by leveraging on advanced technologies like the MISPA I3 that we have spoken about. Artificial intelligence and decentralized point of care testing can improve diagnostic speed, accuracy care, and better outcome, and reducing healthcare costs. Decentralized laboratory, decentralized POCT is a concept where you have all the POC devices in different units within the healthcare system, and even with the patient far away in remote areas or at home. But when you run a centralized POCT unit, basically most of the time due to lack of funding, then you have such a system with such a unit within the healthcare system, and then everybody is consulting such a singular unit, maybe with according to challenges in Nigerian diagnostics. And we have that problem of inadequate infrastructure. I mean, not just the building now, the equipment and the high-tech diagnostic tools. There's workforce and brain drain, which they call the JAPA syndrome now in the country. There is a lot of financial constraint, is highly fragmented to the point that you cannot even gather data. Sometimes when policies come out, you wonder where did they get the data to form such a policy. There are also geographical disparities, which shows quite a long effect on those in the rural areas and remote areas that does not affect those in the urban centers. Well, there are transformative solutions that can solve this problem. Also, improve accuracy and speed, enhance accessibility. I've talked about POCT and telehealth, that's telemedicine. If the POCT system is decentralized well, there is a clinical pathologist in the US who told me that she controls 1,000 glucometers centrally, a single person. But those glucometers are in different remote places, and they are well controlled by telemedicine and telehealth system, which we also need to look into and imbibe. There is personalized medicine. Actually, the era of P4 medicine, since around 2010, 2012, 2013, there was a conference in South Africa, unifying pathology in the era of P4 medicine. That's preventive, predictive, personalized, and participatory medicine that look at individual variability and then is able to help individual patients, not just approaching everybody in the same way. There will be increased efficiency and reduced costs, both from the patient and the health system themselves. There will be better disease surveillance in case of outbreaks, and our policies will then be data-driven, not just forming policies that are baseless without any data support. Now, smarter diagnosis as a concept, what does it mean? It means the integration of advanced technologies, artificial intelligence, machine learning, biomarkers, rapid testing, data analytics. Mr. Sanjay talked about predictive medicine, that's predictive analytics, to provide faster, more accurate, and more accessible diagnostic tool. And the key pillars of smarter diagnosis are listed. Now, when the patient has a illness and comes to the hospital, there are a lot of uncertainties in the mind of the patient. And also the clinicians also have their own uncertainties. And so at the first point, there is experience of the health challenge. And then he comes to the hospital or she comes to the hospital or to see the healthcare provider. They gather information and treat. And when the laboratory all comes to the clinician from the laboratory, there is quite a lot of uncertainty. Too. The clinician might feel that on this test result, do I need to contact another laboratory? Do I need to retest in 2 other laboratories and compare the result I'm getting from this particular laboratory? So all those are evident uncertainty that run through. Now, delayed diagnosis simply means that there is an unwarranted delay or phase lag between the onset of symptoms felt by the patient and then the confirmation of a specific illness condition or health condition that will form appropriate treatment. So once that cannot be achieved, there will be delay in diagnosis and they have deleterious effects eventually. That diagnosis is different from misdiagnosis, where the concern is incorrectly identified or wrongly diagnosed. Delays can stem from various factors: healthcare problem, insufficient screening, lack of funding, people have to travel from far places, communication gap between the patient and the provider. And when we understand all this background, we'll be able to look for potential solutions as we go in the journey. Common conditions that are affected by diagnostic delay: diseases like diabetes mellitus, cancers, and autoimmune disorders, because some of them have subtle signs and symptoms that can be overlooked. And when it's now diagnosed, eventually it can have reached a late stage that the treatment options available might not be able to take care of the condition. And similarly, the silent nature of conditions like hypertension, diabetes result in significant damage even before the disease is discovered, like hypertension, diabetic nephropathy, diabetic retinopathy, and the vasculopathy that may come from diabetes. What are the impacts on the patient and the healthcare system? We have worsened health outcome when the diagnosis is delayed, because it goes on and progressing and nothing is done because there's no specific diagnosis yet. There will be increased complications as a result of the diagnosis. There is reduced treatment option eventually. Even when the diagnosis is now made, the delay and the time wasted will impact greatly on the treatment opportunities available for such patients, and the patients suffer long. There is psychological and emotional depression, and the burden decreases the quality of life eventually. On the healthcare system, there is high mortality rates. It's been said that 30% of deaths that occur in our healthcare system are due to preventable deaths, and there's something we have to do about it. There's unnecessary medical procedures that is the impact of delayed diagnosis, increased healthcare costs, both on the patient and the healthcare system, and there are legal implications also, and the healthcare system can equally lose their reputation if they ever had anyone before. I'm going to, hello? Hello?
We can hear you, sir.
We can hear you.
All right. Okay. I brought up these 4 simple cases to illustrate what I mean by delayed diagnosis, or to illustrate the examples of delayed diagnosis. The first case is a case of AMI, acute myocardial infarction, a 60-year-old man which suffered an heart attack and was taken to our ICU in my center. But apart from the very first matter of AMI, the clinical symptoms, the signs, and everything, we could not do the cardiac markers at that year, some years ago. So troponin was not available, troponin high, troponin T, D-dimer, CK, available. And so we sent the sample to a private laboratory in Nigeria. And I think the center in Nigeria, the center of that private laboratory in Nigeria also would not at that time run the test here in Nigeria. They also have to send it out. So the patient in question managed to survive the ICU care from there. And before we got the result of our cardiac marker, the patient went to his office after surviving ICU admission and had a second attack and passed on to the grave beyond. Such a patient was not supposed to die if everything was available and if there was no delay diagnosis. He would have been treated promptly if the diagnosis was made earlier. Now, the second case shows the power of early detection and pains and penury of late discovery. And we go from biopsy to biomarkers in management of malignancies. So it's a case of suspected malignancy, 64-year-old woman with diagnostic sac. I put the sac there because this woman came from another state to Osun State to my place. Carbohydrate antigen 125, a marker of ovarian tumor, which was minimally above the upper reference limit. The reference limit is 0 to 35 equivalent per mil, or which other unit you want to use. And the result came out to be just 5 above the upper reference limit. So the result was 40. So when the surgeon saw the woman, there was obvious, overt, very fast stigmata of a malignant process going on within the systemic milieu of the woman, but that CA125 result did not show anything. So they went on doing barrage of tests, MRI, CT, endoscopy, this and that. They just went on. It was after a long while that the surgeon contacted me and said, Dr. Adedeji, this CA125 result that this woman brought from another state, can we retest it? That has taken a long time. And I said, yes, let's retest it and see what is happening. To our surprise, we put the sample on our 411 spectrochemical, and the result came out against 40 that the woman brought from outside. At that point, the following day, for laparoscopy, diagnostic laparoscopy, and they picked the ovarian, and histology confirmed it, but it was already late, and the woman gone to the grave beyond as a result of this. The third case is a case of 39-year-old hospital. Unfortunately, was to review a patient at the emergency early in the morning. She drove herself to the emergency, and to come out of the car, she collapsed on the steering. Right there, she was taken to ICU, intensive care unit, and eventually, within 30 minutes of hearing the story, we have run CKMB, troponin T, troponin I, D-dimer, and myoglobin. Was discovered this is acute MI, and of course, prompt treatment followed. So this particular lady in question was able to survive the incidents, and of course, eventually went for interventional cardiology outside the country, and is still alive until today. Such is the effect of availability of equipment and prompt diagnosis. A case of AMI in a 73-year-old. This was an old man who suffered MI, and the diagnosis was made promptly, and we kept on monitoring while they were treating. The treatment was also prompt, and eventually he also survived. So these 4 cases shows us that the outcome, the treatment outcome in each patient depends on how early the diagnosis is made and how prompt the treatment is also instituted. And so the power of early detection, moving from biopsy to biomarkers in the case of the woman with CA125 results, and the smarter solution liquid biopsy and AI-powered biomarker analytics, image analysis for rapid non-invasive screening and monitoring. The impact is early stage intervention, personalized treatment, and reduced patient stress and fast time to treatment. Findings or report have shown that in Nigeria, 2 to 4 weeks is counted before a patient can get some of the results for the investigation. There is 30% increase in mortality, and there's 40% patient abandoning their treatment. And of course, we know the end point that they have to suffer as a result of delayed diagnosis. Existing bottlenecks between laboratory and clinicians. We have shortage of workforce. Pre-analytical errors take about 70%, generally speaking, is being reported. Slow turnaround time because things are not just available everywhere. And there is fragmented data management. Some of the data are available in hard copy, some where you have EMR, some are still on hard copy, some on electronic medical records. Now, there are intelligent solutions to this problem: automated workflows with streamlined laboratory process, and AI-powered analysis, that is things like predictive analytics, can enhance diagnostic accuracy, and integrated data platform where you unify all the data available from health record, history, and AI can even generate summaries from all the things available on the system, where it makes the work easier and the physician or clinician can face their main duty. How intelligent diagnosis enable fact-driven decisions? We have point-of-care testing, AI-powered diagnostics, there's telemedicine and remote monitoring. In some centers, a patient doesn't need to come to this hospital regularly or go to the healthcare provider as often as needed. And they send it to a cloud link with the healthcare provider, and the healthcare provider can manage as many people at the same time. That AI data from various sources and the possible occurrence of an outbreak, disaster as related to health respond more quickly. If we are able to integrate this smarter diagnosis, Nigeria can move from a system plagued by delays to one that provides timely, accurate, and accessible. So this chart shows the through intelligent diagnosis. So sample is available, processing is done on automated system, and there's AI-powered interpretation comparing the result with millions of other available results that have been imputed into the system and reporting a real-time digital result. And of course, decision can be made and clinical integration can be done. This is what we have when we have data-driven decisions, that we have 60% cost reduction, there's 95% accuracy, and the diagnostic time is also reduced. Collaboration as a catalyst for national healthcare progress, there's integrated care delivery, optimizing resource allocation, and driving innovation and improving overall population health outcome. It's going to be a national effect if we are able to look into it. There are mechanisms and principles of collaborating with one another. There will be integrated patient care, that is, you have nurses, doctors, medical laboratory scientists, technicians, the social workers, and everybody combining their effort together for the care of a particular patient in a personalized and participatory way. There will be optimization of available resources. They will be able to share data, share resources, and every other thing available can be shared with understanding. There will be an enhanced public health response, and policy change can then be effective and be effected. Driving innovation and research when there's public-private partnership in the healthcare system and is well streamlined and well formed, then there will be that advantage of driving innovation and research. There's the workforce sustainability, because when the system takes care of the workers will also want to take care of the system. There are factors, however, that are critical for success. There will be shared vision and trust, not mistrust. There will be clear spelled out roles and responsibilities, so that there won't be confusion of roles. There won't be confusion of responsibilities. Data and information are be, and there will be supportive infrastructure and leadership. Now, collaboration as a catalyst have been tested in some settings, and we can have 75 stakeholder engagement, about 200 and above partnership networks, and 5,000 and more health workers can be trained, and you have facilities being operated, more than 150 of them. And such is what we have when we are using the smarter diagnostic approach. In this year, AI in Nigeria, and generally speaking, have been reported to achieve diagnostic support. AI agents in radiology and detecting over 95% of cancer or malignancy at accurate rate. There is administrative automation, that is, human beings do not need to engage in routine scheduling and billing. They can be handled by intelligent system, and that will be a smarter method, and the output also will speak to it. Clinical decision support, real-time recommendation during patient consent can be made right there when we are using AI-powered system. And of course, operation efficiency, 30% reduction in waiting times, and of course, diagnostic and treatment modules can be increased as much as 60 to 70%. This is the summary of what I've just said there as it is. Now, as we go into 2026, 2027, 28 to 2030, there are projections, and in well-planned places, there are plans ongoing and established that by 2028, 20 to 30% reduction of operational costs will be achieved, 50% decrease in supply shortages, and 90% improvement in the bed utilization within the health facilities, and 60% reduction in patient wait times. Every naira that we then save from this can be invested in better patient care and treatment. There's also a projection by 2035 that clinical documentation AI will be able to draft the documentation, gather information from the available laboratory results, the X-rays, the MRIs, the clinical issues have been documented, and refine every information available to bring out the nitty-gritty and the meat of such information, and then can be applied to patient care directly. There'll be operational excellence, and that will also be a great advantage. The 2026 imperative that we are talking about in this lecture is advancing with intelligent diagnostic system. The 2026 imperative for advancing with intelligent diagnostic system refers to the urgent need for healthcare industry in Nigeria to strategically adopt artificial intelligence, because it's human AI cooperation, not that AI will take over the job of a man. There is no fear about it. So it will improve accuracy, efficiency, and personalization of patient care. AI is transiting from emerging technology to an essential component of modern health globally. And this will drive the ability to address workforce shortages and financial pressures and all other challenges that we currently have. The imperative and life necessity for organizations to adopt AI and to remain competitive and to keep abreast of the current event that is being adopted globally. So we have some breakthroughs shaping the future of AI in healthcare and in diagnostics for 2026. There are 7 of them, but I've only picked 2 here, which is applicable to diagnostics. So computer vision has changed from a research method or key to AI healthcare innovation. Computer vision enables AI systems to perceive and comprehend images and videos and then come out with a refined output. It plays a vital role now in clinical practice, and the systems are no longer just extra tool. They are part of the system and they have been clinically validated. So it includes CT scan, MRI, X-rays, and pathological slides that will spot patterns in the odd signs linked to cancer, heart disease, and other diseases that are plaguing the populace. The strength of this system comes from 3 main features. They can recognize pattern, the speed is fascinating, and of course, there is consistency because massive data is input into the system, and there is no now new occurrence that will come up that cannot be detected by comparing with already existing data within the integrated system. Predictive analytics is the second one, and precision forecasting, which Mr. Sanjay talked about, to predict failure, future event, or hazard. Predictive analysis uses historical data, that is, data have been gathered all along for a long time, can be put together and be incorporated into the system, and this can be used for risk stratification, population health forecasting, personalized treatment outcome prediction, operational optimization, and of course, they will be updated continually as new data comes in, because data are now entering the system automatically as the data is being generated from the laboratory. They enter into the system and they are incorporated into the available data and refined and produce new outputs, and it's an ongoing process in the system. This is the building foundation for 2026: infrastructure, technology, workforce, and integration. Now, if we have had the opportunity of interacting with an AI system because of a particular challenge, and I discovered that it's garbage in, garbage out, because what was put into that system could not address my own challenges at that time, because they did not impute the challenge I had. So eventually we went on through the cycles on and on until I felt like putting there, you are not effective, which eventually I sent mails to the vendor and I told them this is not imputed into your AI system and it needs to be imputed because it wasn't captured, and so it couldn't solve my own particular challenge at that point in time. But we have this case study from John Hopkins Hospital and Microsoft Azure. They collaborated and they leveraged on the massive data for medical imaging and genomic information that's available in John Hopkins, and algorithms were trained to predict patient outcomes, disease progression, and read mission risk and respond to treatment in a projective manner. And the implementation of the AI-driven product analytics significantly improved patient care. That was the finding. And this was done by analyzing historical data and identifying ongoing patterns. And the algorithm were able to forecast patient trajectories with high accuracy, very high accuracy, that enabled the healthcare providers in that system to intervene early and prevent complications and make patients to live better life. This was tailored to treatment-based individual patient profiles, that is personalized medicine. The case here demonstrates the potential of AI diagnostics to transform healthcare from reactive to proactive with AI-powered predictive analytics. Well, going from here to 2020, 2026, 2030, 2035, there is the multimodal AI, which gather health systems and unify it and apply it to patients. The way there is also the federated learning, which is a form of privacy-conscious system that we made the information from each healthcare system to be domiciled, but we have a loop in between the contributing and collaborating healthcare system whenever the data is needed, the data is used, but the data remain domicile in the different health systems. So it's able to cover the challenge of privacy. There is also the digital ecosystem, and that federated learning can cover a whole region, like let's say Southwest hospitals in Nigeria. You can cover everything like that. There is also the digital ecosystem that cover a wider area. So if we could start from somewhere like this MISPA I3, if different institutions can imbibe and buy into it, and then we see how it was, and then we progress from there, maybe within your own state, few hospitals in the northern region, few hospitals, and then it extends, then it can become a national offense. So at this point, I want to say thank you very much for listening. Thank you.
Thank you very much for that wonderful presentation. I'll quickly just talk about the product, the MISPA I3. The MISPA I3 is a specific protein analyzer that is cartridge-based. It is used for protein analysis. So earlier, Mr. Sanjay talked about some of the parameters that the MISPA I3 can detect. So the MISPA I3 is important in preventive healthcare. It's important in diagnosis, important also in medication decisions, and also enhances prognosis monitoring. So right from the point of preventive healthcare to the process of monitoring how well your treatment is going, the MISPA I3 can fit into any of these spaces. So it is important, it fits perfectly into the Nigeria workflow system because first of all, one of the major challenges we have in the healthcare space is when it comes to non-routine testing. For instance, like your CRP, your rheumatoid factor, these are non-routine samples for most laboratories, and they may have just maybe one sample. So they may have just one sample or two, and then you have to batch these tests, increasing your turnaround time to patients. But with your MISPA I3, it is cartridge-based. So this means that you can run just one test. As it comes, you run your one test and you give your patient your results. So it takes away the idea of batching to run your samples, reducing your turnaround time significantly. It requires very minimum training because the system is easy to use. And then it gives you guidelines on how to operate the system and how to run the different tests that it can handle. And then when it comes to operation, your MISPA I3 fits into any type of laboratory, from small to medium to large. It is complementary to both your semi-automated analyzers and also your fully automated analyzers. One of the challenges that most laboratories that use semi-automation or manual method will have is how do they run this test? Like your HbA1c or your rheumatoid factor, even your CRP, this test cannot be run on majority of semi-automated analyzers we have in the Nigerian market. And running it manually is cumbersome. Even on your fully automated analyzers, it increases your cost per test when you have to run only one HbA1c sample or one rheumatoid sample or one ASO sample. So what happens? How do you solve this challenge? This is where the MISPA I3 comes in. It will fit into your small laboratory. It fits into your lab, your big laboratories. You can run one sample at a time. And the beauty of this analyzer is that it is compact and will fit into any laboratory space. You don't necessarily have to have a big space to put your MISPA I3. It is very, very small, very compact, but yet powerful. And so looking at the core features of your MISPA I3, top of it is the high sensitivity and strong linearity of this analyzer. So it works on both principles of nephelometry and turbidimetry. So what happens is that this increases your sensitivity for your running. So it means that for samples with very low concentration or samples with very high concentration, that on a normal day will require dilution, right? Your MISPA I3 is sensitive enough to pick this to pick the concentration of this of that patient sample. So it has a very wide linearity limit and a very high sensitivity. And so Sanjay talked about the proton detection, so I'm not going to go too deep into it. The MISPA i3 has an algorithm that can detect your proton effects. So when there is a false negative result because of excessive antigen on antibody in a particular sample, giving you a false negative result, your MISPA i3 has the ability to detect that proton effect, or detect the proton or your hook effect. And then when it comes to your calibration, right, for your HbA1c, calibration is cumbersome because it's a multi-point calibration. MISPA i3 removes these errors, the errors associated with manual calibration. First of all, preparing your calibrators or your standards and all of that. It removes the errors associated with manual calibration, because in your smart calibrator card, the calibration information has already been embedded into that card. So what happens is that once you slot in your card into the analyzer, it transfers that information in your smart card to the analyzer. And so with the smart card calibration system, it is specific to a particular lot and also specific to a particular reagent. So each reagent has its own calibration card. Each lot has its own calibration card. And so on cost efficiency and power efficiency, your MISPA i3 consumes very small sample. Your sample consumption is as low as 60 microliter, 60 to 80 microliter, yes, depending on the test that you want to run. And the reagents are stable up to expiry. So there's nothing like your open vial stability that is associated with many fully automated systems. For your MISPA i3, your reagents are stable up until expiry, and you have very good expiry on those reagents. You have up to about 9 months to about 12 months expiry. And then the beauty of it is that your reagent comes in different pack sizes, right? You have your 10 tests, you have your 30 tests, you have your 60 test pack sizes. So it is depending on what category your laboratory falls into. So if you do not have like a high throughput, you can go for the 10 test. And if you have a very high throughput, please, why not go for the 60 test pack? So your reagents are very stable and easy to access. And another beauty of the MISPA i3 is the battery backup system. Please, how many analyzers in Nigeria have a battery backup system, right? The MISPA i3 has a battery backup system that has a battery life of about 1 hour. So even if there is power interruption, or your light goes off, your generator goes off, for the next 1 hour, you can run on your MISPA i3 without fear of your analyzer going off. And so I remembered why during the conversations Mr. Sanjay had with us, he talked about accuracy. Accuracy is one thing that accuracy and precision is one of the key watchwords in diagnostics, right? Most analyzers of this type, most cartridge-based analyzers do not have QC systems in it. So your MISPA i3 comes with both internal and external QC with your LJ charts. So you're sure that your MISPA i3 is on platforms like your EQA platforms like your Biorad and your Recast EQA platforms. So you know that you can compare whatever results you're getting on these analyzers on these EQA platforms. One other thing that stands out about the MISPA i3 is the precision with your that comes with automated mixing. Your MISPA i3 has embedded in it a vortex mixer. So this ensures that your samples are mixed properly and that your samples are mixing properly during run. And then your turnaround time for your test is about 5 minutes. Very short. It takes you less than 5 minutes to get your HbA1c results or your rheumatoid factor results or your apolipoprotein results. So in less than 5 minutes, you are sure that whatever sample you're running on that analyzer, you're going to get your results. And then the machine comes, your reagent packs are barcoded and pre-filled already. So you don't have to do any any mixing or any external preparation. So all of these have been done and embedded in your reagent packs. So we're briefly going to be looking at MISPA i3 versus other platforms, right? Other point of care analyzers, other cartridge-based analyzers, other protein analyzers. So when it comes to the principle, your MISPA i3 has a wider range. So it uses both your nephelometry and your turbidimetry principle. And then your unique channel shifting technology, which allows you to move from either your nephelometry or your turbidimetry, depending on the type of sample that you're running, whether it is low concentration or it is high concentration. Trust me that you have it covered with your MISPA i3. And when it comes to your sensitivity here is high, unlike others that are subject to drifts. For most laboratory grade standards that require that your machines are highly sensitive to be able to provide results for either low or high concentration samples. So your MISPA i3 has a smart algorithm that alerts when there's an excess antigen production. So it also supports your QC, internal and external QC. It's, like I said earlier, it's available on your Recast and your Barrett platforms. So it's able to maintain a steady temperature. It has a temperature control system such that the external temperature of the room, whether it is hot or it is cold, does not necessarily affect, does not affect the temperature of the machine. So the temperature at which your tests are run is uniformly at 37 degrees Celsius. Like I said, your turnaround time is 5 minutes per test, less than 5 minutes for some samples. And your accuracy, you have a coefficient of variation greater than 5%. So now we're just going to briefly look at the different test panels or the different parameters that your MISPA i3 can run. So you see, we have a wide range of tests that your analyzer can run. On your glycemic control, you have your HbA1c. It can also run your cysteine C and microalbumin, which are important for early kidney disease detection and monitoring. For your inflammation and infection, you have your CRP, you have your high sensitive CRP, you have your procalcitonin, you have your isotiter and ferritin. Under your cardiac metabolic panels, you have, or markers rather, you have your apolipo A1 and apolipo B, and then you also have your lipoprotein. You have different immunoglobulins from IgE to IgG, then your complement C proteins. And newly introduced is your free kappa and your lambda light chains that are important in myeloma detection. So you also have your vitamin D and prealbumin as part of your markers for your bone and nutritional health. So we've talked about the MISPA i3, we've talked about the benefits of the MISPA i3. And so we're pleased to inform you that there's going to be a discount on your MISPA i3 for the next 2 weeks post-webinar. So you have a 30% discount on your equipment and a 5% discount on reagent. So please take a screenshot or take a picture of this number and send in your inquiries. We're happy to even help you customize the solution to fit to your laboratory. If there are other testing that you want to, that you're not currently running in your laboratory that you think that you can do, we are ready to customize different solutions as it fits to your laboratory. So take a picture of the number, and the discount lasts for just 2 weeks. Grab your copy now. Thank you very much. So that is that on our MISPA i3. And please don't forget to drop your questions and comments in the chat box. We'll be happy to attend to it as soon as we have time for that. And also we have one of our end users on the call, person of Mrs. Tony. She uses our MISPA i3 for various tests in her laboratory. And so we'd like her to just come up and give us a brief 2 minutes testimonial on how her experience with the MISPA I3 is going. So, Ms. Toyin, the floor is yours.
Good afternoon, everyone. Thank you, Kemi, for inviting me to this very educative and rather enlightening webinar. So my name is Olugboko Oluwatoyin. I'm one of the end user of Inspire 3 at the present moment. And in my expertise and in my experience so far, as regards machines like this, I can actually say, categorically say, Inspire 3 is one of which stands out. We all as end users of this type of machines, chemistry machines, we know how difficult it is to actually maintain and keep HbA1c, HbA1c, CRP, and rheumatoid factor running on our major machines. So that was one of my major concerns when I said I needed a separate machine that can run rheumatoid factor, CRP, and HbA1c. And we've been using this machine like now for a year, and we've not had any downtime, and our result has been coming good. Another thing that is of good to this machine is it has a control, not expensive, and we have the normal and the pathological. We run our controls alongside. We are currently subscribing to EQA because we're trying to go for ISO for the same machine, and we have passed the last two, so the first two so far. So I can say that the Mispire 3 is actually up to what they are saying. So I think that's all I can say for now. And again, he was right when he said no wastages, no rerunning of tests. You can actually say for 60 tests, you can actually say you were able to use it to run 60 tests. So far, you follow the instructions and maintenance of the machine, which is not something so difficult to do. So thank you very much.
Thank you very much, Ms. Tony, for that. So at least you've heard from an end user. It's not just us trying to sell a machine. You've had her experiences with the MISPA I3. So it's time to grab your copy now while the discount is still available. So please, does anyone have any questions? So someone asked if the slides will be shared. So the slides will not be shared, but the video of this webinar is going to be uploaded on YouTube. So I think I'll share a link to everyone or the participants of this webinar.
Someone is asking about the price of the analyzer and the reagents.
Oh, okay, sorry. The price of the analyzer is 4 million. So with your reagents, right, depending on, like I said, you have different pack sizes, 10 tests, 30 tests, and 60 tests. And it's also dependent on the parameter. But trust me, for your HbA1c, your cost per test is as low as 2,500, and it even go lower. So the more packs, the more tests you get, the lesser your reagent cost. So the cost of the analyzer itself is 4 million.
I have a question.
Okay, I can hear you.
Is that not a way maybe for placement? And there be placement of the equipment that will not be buying reagents? Is it possible?
Okay, so with placements is a very tricky option. It really depends on your workload. So if you have very good workload, yes, we're ready to talk about placement options. Okay, so I think I can invite Mr. Elijah for the closing remarks. Mr. Elijah.
Okay, so thank you everyone for joining. Distinguished colleagues, esteemed participants, I appreciate your time. So as we conclude today's highly insightful scientific session on Agappe MISPA I3 protein analyzer, I would like to briefly highlight the broader diagnostic value that Uni-medical brings to laboratory and healthcare institutions across Nigeria. Now, at Uni-medical, IVD department, by the way, we have other departments, but specifically IVD departments, our commitment is not only to introduce innovative analyzers like the MISPA I3 that we just talked about, but to provide complete integrated diagnostic solution that enhances value, accuracy, efficiency, and clinical decision making. So beyond protein analysis, Our portfolio also covers full spectrum of laboratory diagnosis, from immunoassay to clinical chemistry, where we represent Agappe, hematology 3 and 5 parts, where we also represent Agappe, urine analyzer, coagulation analyzer, these protein analyzer that we just talked about, and labeling analyzer. These are all from Agappe, right? This is our Agappe portfolio, which ensures that every lab, regardless of the size or complexity, can access world-class latest technology aligned with global standard and practices. Then our immunoassay analyzers, we represent SNIPE, and we have several series which is best or can be best fitted or suited for your laboratory size as well, medium, big, or small. It is fully automated with the broadest test menu and segmented test pack size. I wouldn't want to dwell more on this, but I just want to say that beyond the protein analyzer that we centered our webinar on, we have other analyzers from Agappe, from SNIPE, and from Sysmex. So together, this system creates comprehensive, what's the word now? Comprehensive diagnostic ecosystem supported by our nationwide team of sales and application specialists and service engineers who ensure seamless installation, training, validation, post-sales technical support. As we speak, one unit of Mr. I3 was installed yesterday in Abuja, right? And the end user, I spoke to him this morning. He gave an amazing testimony on how the installation and training went. So we hope that in the next couple of weeks, and taking advantage of the promo that Kemi have told us about or informed us, 2 weeks promo time frame, we hope that we are able to have larger installation. We hope that we are able to bring value to your lab when it comes to protein analyzer. So as we close again, I want to express appreciation for your active engagement. We have a total of 54, alternating between 54 to 58 thereabouts. We really appreciate this. We invite your facilities to explore how Uni-medical can partner with you to upgrade your diagnostic capacity, reduce turnaround time, improve patient outcomes, and strengthen your laboratory efficiency using reliable, efficient, scalable instruments suited for the Nigerian healthcare environment. Thank you once again for joining us, and we look forward to supporting your laboratory needs with solution that delivers value, accuracy, and long-term performance. I appreciate everyone for joining us. Thank you.
Thank you everyone for joining our call. Please don't forget to follow us on social media, on all our platforms. It was nice having everyone on this call, and I look forward to meeting with us physically. Thank you so much. Have a nice day and enjoy your afternoon.